First of all, I'd like to thank Jerry Pollack for this very kind invitation to present our work here in Bulgaria and the organizing committee. This is my very first presentation in Bulgaria. I've given at least 12 in your neighboring country, Romania, on several occasions, so it's very nice to be here. So the topics that I want to talk about in the 20 minutes that I have are shown on this slide. First of all, I'd like to introduce why we're interested in water. And this will take us into the world of theranostics, which is defined as a combination of medical therapy with detection. And this will lead me to an issue that's been a problem with respect to biomedical devices for many long years. That is the interaction between devices and the biological fluids like blood and urine and tissue. And I'll briefly comment upon the role between fouling and biocompatibility. Our work, we're very interested in antithrombotic surface chemistry, and this will come down to surface hydration, which is really the purpose of the connection to this particular symposium. And I'll talk a little bit about the study of this hydration using neutron reflectometry, and I'll make some questions, set the audience some questions at the end on what you hear today. So we're very interested in treating very seriously ill patients. This astonishing array of equipment is what you have when you have patients in intensive care, and our particular interest has been in septic shock. We're not particularly interested necessarily in inflammation and coagulopathy, but we are interested in shock. And the theranostics is a way of dealing with that. And in theranostics, we take blood from a patient, we pump it through a special cartridge to remove offending molecules from the blood. And there are a number of strategies for doing that. For example, in septic shock, we remove molecules like lipopolysaccharide, which is endotoxin. And then we also like to detect those molecules in the blood, which is then passed back to the patient. So this is a kind of a circular system, which is combining detection with therapy. And that's effectively the core of our research in our lab. So one of the things we do is we produce various types of sensor. And one of the ones we use is an electromagnetic high-frequency acoustic sensor. In this particular case, we have a piezoelectric device here, which is detecting surface chemistry and detecting the molecules in the blood or serum. We flow serum over the surface, and we can detect it. And this sort of detection system would be incorporated into the theranostic circular device that we saw a moment ago. Now, if we do that, carry out that experiment with biological fluids such as serum, which contains, of course, a whole range of different molecules, and this is the sensor frequency that we're actually measuring, and this is time. As you flow serum over the surface of this device, you end up getting a very, very, very large signal of some 34,000 hertz, 34 kilohertz. And that signal is caused by molecules from serum attaching to the surface. In this case, it's actually quartz, a quartz device. Now, we have produced surface chemistry in the last few years in an attempt to avoid all those molecules attaching to the surface. And what we produce is bifunctional silane molecules. The trichlorosilane functionality attaches to the substrate, as we see here. And then we have a chemical backbone which contains an oligoethylene functionality. And then we can attach various molecules to that surface, for example, to make a biosensor. This shows you the kind of surface chemistry that lies behind that, which was published a couple of years ago. And it's this particular key molecular system that I want to talk about here today in Bulgaria. This particular molecule on a surface is only about 5 angstroms thick. It's very, 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 very thin layer. As you can see, it has an oxygen, ether oxygen in the middle, and an OH group, functional group at the end. And I'll show you in a moment that this molecule possesses some very interesting properties in terms of hydration of this surface. Now, if we carry out that same experiment that I showed you a moment ago, remember we were looking at bare quartz with our sensor, which has a 30-some thousand hertz shift in value here. This particular molecule here, you see that the fouling of the surface is reduced down extremely significantly when this molecule is attached to a surface. And that is actually a really quite remarkable result in the sense that you've reduced down surface fouling by more than an order of magnitude here on the surface. So this molecule, in this particular case on quartz, has very, very remarkable properties. Now, some time ago, a couple of years ago, we were discussing these results with people in the world of biocompatibility, not so much in the world of sensor and biosensor technology. And their particular interest, of course, is what happens when blood contacts a number of medical devices. And medical devices, of course, you have many types and structures there. You have stents, you have circuitry, you have renal dialysis for patients with renal failure, vascular grafts and pacemakers. This is not a comprehensive list, but it shows you the kind of things that people are interested in when blood contacts these various devices. One of these types of interaction is in what is called bypass circuitry. Very often when guys have a heart attack problem, they have to have what's something called bypass surgery, where they reroute connections in the heart. You see here one of these operations taking place. And one of the issues here is that many of these materials are actually made from from polymers, or polymeric materials. But although these surgeries are often very successful, some years later, 5 years later, 7 to 8 years later, these patients almost universally have brain disability problems. They have the clots find their way to the brain, and these cause serious medical issues. So yes, the heart problem is fixed, so to speak, but they get problems later on. And this is certainly the case, for example, with folks who have renal dialysis. We have a very large audience here, and there will surely be somebody in this audience who has to have dialysis 3 times a week. And renal failure has a very, very poor prognosis in terms of the future. And it's connected to what actually happens when blood interacts with these surfaces. So the issue was, can we use the surface chemistry that I've talked about already, and can we put that onto these kind of materials and see what it does in terms of antithrombogenicity. So here is the nature of the experiment. Basically what we do here is we flow blood. It's usually blood from a graduate student. They donate and usually faint on the prospect of doing so. I've only given blood once for this, and you have to give about 100 mils for this, but now we've got it down to 2 mils, fortunately, so you can prick your finger virtually. But we flow blood over a surface of the coated polymer that I've talked about, and we have an inverted fluorescence microscope here which actually detects. What we actually do is we take platelets in the blood and we label these with a fluorescent agent so we can actually see what's actually happening in real time on the surface of these materials. And here is what actually happens when you have one of these surfaces, and this is blood which is flowed over, a still picture of blood flowed over a surface on the polymer polycarbonate with collagen coating. As you can see, as our American friends would tell you, this lights up like the 4th of July there. And what's actually happening is these are aggregates of platelets on the surface, and you certainly don't want that in your bloodstream, which is going to find the brain and cause issues. So here is what actually happens with the bare polymer. You see these aggregates here. This is a few minutes experiment with blood flowing over just a bare polymer, the sort of thing you would use in a medical device. And here is the system where you have that molecule I've been showing you coated onto polycarbonate. It stops thrombus formation, these particles, are on the surface by 98%. There's a huge reduction in aggregate formation on these surfaces. And actually that is far, far superior to other types of coating system which have been used over many years in the literature. So one of the things that we wanted to do was to study that, which is very relevant to this particular audience and this conference, and I'd very much appreciate comments upon the nature of what water is doing here. We've long felt that this is caused by hydration of these very thin films. And remember that these films are only 5 angstroms thick. They really are ultra thin. So the role of water, either from the bulk or very close to these surfaces, is clearly quite crucial. And of course, studying what water does in a 5 angstrom film is a difficult task. We've heard a lot today from various speakers about organized and structured water. One of the techniques that we have used extensively is neutron reflectometry. We're very fortunate in Canada to have a very good neutron beam research center, which is in northern Ontario. It's about 600 miles from Toronto, north direction. And basically, we can get very close to a nuclear reactor, and we can study a whole bunch of materials. Our particular interest is in using specular reflectivity system, looking at interfaces. So this is a very ideal, although expensive experiment to conduct on these surfaces. And so basically, there's an awful lot of physics on this slide, which I don't want to go into, but basically what we record is reflectivity against distances from the surface using something called Kiesick fringes. And so basically from a very detailed analysis of the physics of how neutrons interact with this hydrated surface, we can look at the actual structure. And basically, so we can model these things. This is bare silicon. This is this molecule that we've been talking about here. And when we actually look at that model, we can produce reflectivity fits. We can actually fit down. We have computer software and calculations which can actually draw up various models for this. So we have to put this data into the system. And when we do that, what we come up with, which would be more interest to this community here, is we can actually describe what the water actually looks like. And what's important, what comes out of this, is that on this particular molecule, which I've been showing in the lecture here, there are different types of water. There's water intrinsically incorporated in here, which very much has the type of structure that many of my colleagues here have been speaking about at this conference. It's a structured water. What the precise nature of that structured water is, we will not be sure, but one thing is for sure is that neutron reflectometry is actually seeing that. And also, as you move out into the liquid here, there's another different type of water, and then we find ourselves in the bulk. So to cut a long story short here, this particular configuration here is actually conferring remarkable biocompatible properties on whatever material. In this case, it's quartz, but of course this is polymers, as I've mentioned, and indeed right now we're looking at things like stainless steel and other types of materials such as aluminum nitride that give you the same behavior. So hydration and what water is doing in these extremely thin layers of 5-6 angstroms here is actually crucial to the world of biocompatibility. We've also carried out a number of molecular dynamic calculations of this system, which I'm not going to talk about because my distinguished colleague Sonia Sheikh from my lab is here with us at the conference, and she has a poster about 10 meters outside the door there which describes this work. So I'm sure she can talk about it tomorrow night with the group here. But I want to finish with some questions here. This morning, Jerry Pollack talked about vested interest and entrenched views and mantras and dogmas is what he was talking about. There is a myth in the literature that very high quality self-assembled monolayers reduce adsorption and fouling. This turns out to be completely invalid. And if you try to publish the kind of work that we have published here, we met an awful lot of resistance. In fact, it's been worse than resistance. It's been downright vitriol. We've actually been criticized extremely severely, despite the fact the results here are shown are actually extremely valid. So I know what Jerry's talking about by vested interest. And vested interest in this technology is enormous in the world of nanotechnology. None of it's nano, but they call it nanotechnology. Nanotechnology used to be called colloid science about 40 years ago, but to get big research grants now, you call yourself a nano specialist. So I know what Jerry's talking about here. There is the issue here of what is the precise role of that distal OH in the molecules that I've been discussing, and by that question I'm implying that we don't understand the full implication of the water structure that's there. Thirdly here, it turns out that it's not so critical how we make our films. You would imagine that it would be very critical in terms of setting up this water structure. It turns out that it's not so critical, but I'm using the word robust here. So why is that? Why is it that these ultra-thin layers may not be prepared so carefully, but they seem to be very consistent in their behavior? So that water structure or hydration layer that there is not critically dependent upon necessarily on the molecular conformation there. That's a question. Can this surface chemistry make huge shifts in the way that people do surgery? Can we avoid the medical conditions in the future that I've referred to in my talk here? Can we now produce medical devices that do not cause cognitive disability after 5 or 7 or 8 years? Is that possible? Who knows? But that is a question. And here is the last point I'd like to make is hydration in membrane surfaces and protein chemistry, of course, has been recognized for many years. But in terms of the selectivity of biochemical reactions, such as involved in cell signaling and antibody-antigen reactions, it's usually ignored. So only really discussed in terms of entropic considerations. But maybe the structure of the water is playing a part here, which is usually discounted entirely. So I'd like to pose that question. Are the results of our work here relevant to a different world entirely? And what is the role, what is such a role of a structured water in biology? This is not something which has been very prevalent in the literature. So I'd like to finish by showing the members of my research group, and in particular, I'd like to mention Sonia, who is, as I mentioned, who is here. You should all congratulate Sonia because she got her PhD last week, or was it 2 weeks ago now? I can't remember, but it was a couple of weeks. She just did that, based pretty well entirely upon the work that you see here. And our synthetic organic chemist, Chris Blazikowski. These molecules, by the way, that I've showed you here today are often 10, 12, 13-step syntheses, and they are not trivial from a synthetic point of view, and he actually did most of that work. We're a very interdisciplinary group at the University of Toronto. Jack is from Taiwan, and Sonia is originally from Bangladesh. Brian is Canadian. Elaine is from Hong Kong. Kirill is Kazakhstan. Pat is from Italy. Chris is French. Larissa is Romanian, as is Victor. Janice is from Hong Kong. Niall is Irish. Edmund is Hong Kong. These are people from the People's Republic of China. This chap is from India, and these two are from Portugal. So we collectively speak 12 languages in our lab. And actually, we have no problems getting papers translated. What we don't have is anybody from Bulgaria, which I hope is going to change after this symposium. So I'd like to thank you for listening to me, and I'll certainly be happy to answer any questions. This is our CN Tower in Toronto, which is very famous, which is 1,834 feet high. Until about a year ago, it was the world's tallest freestanding structure. But they built a big thing in Dubai, in the Arab Emirates, which is about 1 feet 6 inches taller here. So now they claim to have the world's, but they're going to put a thing on this in a few weeks with a helicopter, which is going to be 6 inches bigger than the Dubai thing. So all the literature that we produced in Toronto and Ontario for tourism, so people from Bulgaria can visit us, they can come and see. There is a restaurant here, by the way, which you can see the United States from there and eat your lunch. It's a fantastic view and a fantastic place to go to. However, I should warn you that the food is absolutely lousy there because they make it here and then it goes up there in an elevator. You can imagine what that does. So I'll finish up there and answer any questions that you might have.